Welcome, everyone. Thank you so much for showing up to another of our Build a Cell seminar series. And thank you. Keith is going to be sharing his research with us. And Keith, I will let you introduce yourself and then dive right in.
Great. Thanks, Chris. Thanks, Kate. Yeah, so thank you to both of you for the chance to share our work today. I'm going to just pull my slides up. All right, so there we go. If anyone has questions, I'm happy to keep it really casual, and you can interrupt, ask questions at any point that you want, or save them to the end, either way, or let me know if you can't hear me or something. So I'm Keith, and today we're going to talk about how we're using cell-free synthetic biology and low-cost hardware to improve access to diagnostics. And I'm from the University of Toronto, and Kate mentioned that I should introduce myself, so I prepared a little tour of how I got here. So I'm from Canada, and I started my academic career at the University of Alberta, where I studied botany. So long ago that this was not even molecular botany. This was like classical botany way back when. But I was fascinated with how small molecules could be used for plants to defend themselves. So these like chemical defenses, chemical biology. So I then went and did my master's at UBC, where I did natural product chemistry with Neil Towers. And I guess the main point of all this is also that my career path has not been a straight path. So I think that's maybe useful to know in your early part of your career, that there's lots of wandering is not necessarily a bad thing. And so I did my master's with Neil, and then I was again fascinated with how could a small molecule regulate the developments or the gene networks of an organism. So I came to the University of Toronto, where I did my PhD in molecular genetics with Aled Edwards and Henry Krauss. And here I did structural biology and functional proteomics, and looking at nuclear receptors, which are drug targets in humans, and working on de-orphaning them using mass spectrometry and structure. And then I changed gears quite a bit and decided that I wanted to translate my training in fundamental biology towards applied biology. And so this is when I went to the Wyss Institute, where I did my postdoc with Jim Collins. And I loved it. And it was sort of career changing. And then I came and started my lab at the University of Toronto in 2016. But this is not a straight path. There were lots of interruptions along the way. One of them was taking 3 years to go to carpentry school and build things. And so I think what we do now in the lab, which is building with proteins and genes and things like that, was really informed by my time building things, actual like physical things. Also, just given the sort of context of this, I thought I'd start out with some unpublished work that I did way back at the very beginning of working with cell-free systems. And so this was really inspired by Vincent Maro's work and some work out of the Netherlands at the time, where I was putting gene circuit-based systems into liposomes, and also into hydrogels and oil water emulsion systems. So this is where it all started. And then over time, I expanded to bring in paper-based systems. So here, I'll talk about this in more detail later, but these are toehold switches, which are activated by trigger RNA in a sequence-specific way. And here, the toehold switches are turning on different colored fluorescent proteins or a colorimetric enzyme. So just those are early things that I did. But now we're shifting over to what the lab does now. So I'm going to start off talking about our fundamental platform. This is a cell-free paper-based platform that we run our function sort of tools in. And then I'm going to talk about our efforts around decentralizing molecular diagnostics and the patient trials for chikungunya and Zika, a glucose meter interface that we published a couple of years ago, and then a new CRISPR-based tool that we published this year, as well as some early work on some reagent production for diagnostics that we also published this year. So themes in the lab, we are really focused on decentralizing access to biotechnology. And perhaps like many of you, if you work with cell-free systems, because they are cell-free, there's no cell wall or membrane in the way, we have this opportunity to put our engineered molecular systems, maybe I'll turn on my pointer. Molecular systems directly with hardware, which we've done, and that's been fun. Another sort of central theme is that we develop new technologies, and we publish a paper, and then we're really focused on taking those technologies into the real world and deploying them and testing them, and that helps inform us on the next challenges. What are those next challenges? And sort of along that sort of theme of translation, we've now had 3 trainee-led startups come out of the lab. And I'm going to tell you about those as we go through the story of the patient trial and how the students involved and how we got to where we are. So as I mentioned, our lab's really focused on decentralizing tools for healthcare. And we do this primarily through cell-free gene circuit-based diagnostics and portable cell-free biomanufacturing. And these are approaches that are relatively new that we and others in the community have been pioneering over the last 10 years. And the main sort of thesis of our work is that if we can make these tools low cost and portable, that we can improve access to healthcare. And the nature of our work means that we have collaborators around the world. So we've got collaborators in India, Latin America, and the US, of course. And a lot of our work's focused in technologies for food security, emergency preparedness, general healthcare, and national security applications. Many of you may be familiar with cell-free systems, but I thought I would just run through it. So we use the cell-free enzymes of transcription and translation, primarily from E. coli, but we could be using these enzymes from plants, mammalian cells, fungi. There's lots of those systems that are out there. We then combine it with DNA encoding our tool. So in this case, this is a sensor. So we have some kind of a synthetic gene network that can detect our analyte of interest. And for the work I'm going to talk about today, that's usually the pathogen's genome in a sequence-specific way. And then there's an output that the user can interpret. Or this could simply be a gene encoding lab reagent or therapeutic. We then freeze dry these so that we can distribute them without refrigeration and store them. And in the sensor sort of modality, we embed the cell-free enzymes of transcription and translation into paper discs. These are 2-millimeter paper discs at the bottom of a 384 well plate. They just fit nicely in there. And the disc on the right also had the DNA encoding for green fluorescent protein. We incubate at 20 to 30 degrees for 20 to 30 minutes, and you start to get fluorescence. And then for the manufacturing side, we do this without paper, right? So these are just freeze-dried pellets. And over time, you get the expression of your protein. Again, this is just GFP for demonstration's sake, but this would be your antibody or your vaccine subunit, etc. So what I just told you about was essentially, you can think of it as the operating system. And now we need to switch to sort of what's the app, what's the program you're going to run on that operating system. And so this work was done in collaboration with Alex Green, who I met through my postdoc with Jim Collins, and Alex was with Jim as well as Peng Yin at the Wyss Institute. And Alex is now at Boston University, and we still are great collaborators. And he developed this toehold switch, which is an RNA hairpin that sits at the 5' end of an mRNA. That's generally the reporter protein. And the loop sequesters the ribosomal binding site and the start codons in the stem. And this prevents the ribosome from binding and translation. And so the way it's designed is this AB region here is essentially the sensor surface. So you put the complement to your pathogen's genome in this AB region. And here we call it the trigger RNA, but would be the pathogen's sequence. And so when A star binds to A, you essentially get this like tab on a zipper, and you unravel the hairpin, you get linearization, ribosome can bind, and you can get translation of your gene of interest. And just the nature of these things, we can largely detect any sequence of interest. But of course, there are other features we had to develop to make this useful out in the real world. So I already mentioned freeze drying. And so that lets us distribute without refrigeration. For low resource settings, we developed color-based outputs so that the user can tell what's going on at the molecular level with their naked eye. And for more sophisticated applications where there's quantification or need for automation, we developed low-cost devices. And so in our first paper that we put out in 2014, we took the toehold switch and embedded it with freeze drying into paper. And we developed this color-based output that you could see with your eye. And Tom Ferrante, who's at the Wyss Institute, was the genius behind developing this reader, which I think was only $30 to build. So this was really exciting at the time. This was the first time you could deploy these sort of gene circuits outside of a cell, so in a biosafe way. But they were not sensitive enough to detect clinically relevant concentrations of genomes or sequences. And so we spent the next year, and Melina Fan from AdGene came and did a sabbatical with us, and she had the bright idea of putting isothermal amplification loops in front of the toehold switch. And so, and right around that point, I came to Toronto to start my lab, at which point the Zika outbreak happened in Latin America. And so Jim Collins phoned Alex and I up, and we agreed that with the addition of the isothermal amplification and several other improvements, we'd sort of move the platform far enough to make a contribution. And so a large team, including us, moved this platform forward, and we were able to detect low attomolar and femtomolar range of the target of these amplicons. So the isothermal amplification makes amplicons, the toehold targets the amplicons. And so with this, we could detect global strains of the Zika virus while distinguishing from other mosquito-borne infections. And then Guillaume Lambert, I think, was the first to use CRISPR in a diagnostic here. This enabled us to discriminate between the American strain of the Zika virus and other global strains. And this was at the time really important because of the neurological issues like microcephaly that were associated with the American strain virus. We were then fortunate to get funded by CIHR and the IDRC, which are Canadian funding agencies, to take our work to Latin America. And these were sort of the 4 leads that have been part of my lab in the last few years. And so we went down, and but there, of course, were many other authors involved. And so I just want to acknowledge all of them in this work. They were central to that. And the main focus of this project was to simply test our paper-based platform with patient samples. And so we teamed up with universities or research centers in Brazil, Colombia, and Ecuador and Alex Green was at ASU and Jim at the Wyss Institute. And so when I wrote this grant, it was one of my first grants, I was not aware of the fact that our partners in Latin America didn't have 384 plate readers, which was really important for the economics of these tests. So small volumes, low cost. And so we ended up having to build our own plate reader. But I was very lucky that I had two super bright master's students, Sarai and Livia, who developed what we call Plum, which is a 20 cubic centimeter plate reader that could go in a suitcase. This is how we got it to Latin America. They just, they can be battery operated. They have a computer on board. We developed the user interface as well, and they could automatically discriminate between positives and negative samples. And we could remote in, and the data was stored on a secure database. And there's just a cutaway of a rendering of the Plum reader and its various parts. And halfway through the project, we sort of realized we'd created something that had more value beyond just this patient trial. And so with Sarai and Livia, we founded LSK, which was, and this was their plate reader, and it was acquired by Nicoya last year. And so that was a great outcome for that. Along the way, we had biologists like Margot building hardware, and we had hardware folks learning molecular biology. So it was a great sort of interdisciplinary effort. This is us in Ecuador with the team there and from other centers. And this is us with Iquiquito in the background on one of the volcanoes nearby. And then the following year, we went to Recife in Brazil, which is the epicenter of the Zika outbreak in Brazil, which is where a large part of the patient trial was done. And so here, when you have a diagnostic, right, some sort of key metrics, one is specificity and the other is sensitivity. And so on the left side of the graph is the data from the Plum reader. So you can see the quantification on the Plum reader here, and you can see by eye here the purple for positive, yellow for negative. And on the right side, we have RT-qPCR. This is using the CDC RT-qPCR for the Zika virus. And you can see that against a panel of other mosquito-borne viruses, we could detect, we were specific for the Zika virus. And of course, PCR was the same. So we were happy with that. Here we have a titration of cultured Zika virus from 10 to the 4 down to 10 to the -3 plaque forming units. And we could detect down to 10 to the 1 with our system. And we were quite pleased that this matched the detection limits of the RT-qPCR assay. And so with that sort of established, we went on and did the patient trial where we looked at 268 patients and found an accuracy of about 98%. And then wanting to sort of demonstrate the extensibility of this concept or platform by simply changing the primers for the isothermal method and the little toehold switch sequence, we were able to point the system at chikungunya, which is another mosquito-borne infection. It's a smaller subset of patients, but we found very similar level of accuracy. And so again, this is Margot's gone on with Alex and Guillaume and myself to start Encarta. And so another trainee-led venture there. Now working in away from home, where you have all of the things you need and all that is a challenging way to do science, but very rewarding, and also has some silver linings. So some of the silver linings are that it really highlights what the technical challenges are over the horizon. So one is multiplexing. So if you have someone who's ill in front of you, clearly something's wrong, but they're not positive for Zika. But you need to figure out so you have to test repeatedly until you get the right, malaria, chikungunya, etc., dengue. So instead of, it would be really nice if you had just one test that tested, 20 things versus a test, 20 separate tests and the unit of cost associated with that. And so that's something that we have been working on, and we've published one paper on that so far. We're not going to talk about that today, just in the interest of time, but we will talk about the other demonstration. So gene circuit-based diagnostics are a fairly new modality, and so really wasn't clear how we could put this capacity into the hands of users. And so for that, we turned to a glucose meter as our sort of like piece of hardware that's already mass produced, super inexpensive, available globally, accepted by clinicians and patients alike. And so this was the bright idea of Evan Amalfitano, who did his PhD in the lab and is now at the National Research Council of Canada. And his idea was to replace in the sort of the gene circuit or toehold switch, replace the enzyme that converts the substrate from yellow to purple, which is beta-galactosidase, to a enzyme that can convert a inert precursor into glucose. And so after screening, I think 40 different enzymes for high-level activity in the cell-free system, right? So under the salts and the pH conditions of the cell-free system, he found tralase, which converts trehalose into two glucose molecules, lactase, which converts lactose into, I think it's galactose and glucose, and a phosphatase that converted glucose 6-phosphate into glucose. And then the idea being that you can then read that glucose signal using this hardware. So you've got some kind of a number. And so to consolidate Evan's PhD down into 1 or 2 slides, we chose to build this diagnostic around a disease that is sort of found in parts of the world where there's limited infrastructure for healthcare and for water treatment. And so typhoid fit the bill. In retrospect, this was a very challenging disease to try to work with. So that added a bit of time to Evan's thesis, but in the end it worked well. So this was using S-typhi, which is the agent that causes typhoid. Evan developed a system that could detect 1,000 colony forming units per mil, which is sort of the known or the current limit of detection using culture-based methods now. And he developed a platform where without extraction, so just centrifugation and then heat-based lysis, followed by dilution and NASBA, which is an isothermal amplification method, and the toehold switch, he could detect typhoid and distinguish from standard. So just to orient you here, on the y-axis is milligrams per deciliter of the glucose that's been generated. And so, as you can imagine, a user is not going to really know the difference between 100 milligrams per deciliter and 200 milligrams per deciliter. And so for that, we developed an app that could go on your phone and convert. You'd put in your sort of control read, followed by your sample read, and then the app would go to a lookup table and tell the user in plain language what the outcome was. And this was wrapping up right around when here. Just lost my screen. Okay, right around when COVID hit, and so of course we went to pivoted to COVID, and so Evan was able to detect again using the glucose meter. So all of this data was collected on an $11 glucose meter. You can see the error bar is really remarkably small, which is a testament to all the engineering that's gone into these glucose meters over the decades. But he could detect down in the sequence, this is synthetic RNA, I think, down to low attomolar range, had specificity against other respiratory infections. So these are 2 influenza strains, and MERS, which is another coronavirus. So we could detect SARS-CoV-2 from that. And then in a very sort of small patient trial, I wouldn't call it a trial, but patient samples, where we had 3 healthy volunteers and 3 COVID positive samples, nasopharyngeal samples, the glucose meter could detect that as well. And so Fahim and Sarai from the lab built this portable incubator that could both heat and cool. So we could switch from the isothermal mode to toe hold switch mode. And this is about the size of a grapefruit and cost, I think, just under $100. And so here, just to sort of show that this incubator works, we've got data collected using a commercial thermocycler with and without trigger or target, and then the portable incubator with and without target. And you can see while our error bars are higher, the general outcome for the average is pretty comparable. Okay, so now we're going to shift to a really interesting CRISPR-based technology that was developed by Margot while she was in the lab, called Repair. And how it differentiates itself from other CRISPR-based diagnostics is that it doesn't require any RNA inputs, which is sort of how the conventional CRISPR-based technologies require these RNA-based reporters or guide RNAs. And also, instead of just relying on collateral cleavage, it will do that, but it also creates protein-based outputs. So that's sort of how this works. And so how the system works is it could detect either RNA or DNA inputs, and then using primers that tacked on a T7 RNA polymerase or promoter for T7, or a primer that contained the CRISPR RNA, so basically a barcode. Using HDA or RT-HDA, this process would essentially barcode the target of interest, the pathogen of interest, and then the Cas proteins could auto-cleave. Then there'd be transcription, I forgot to mention that. And then these Cas proteins could auto-cleave the CRISPR RNA out of that longer RNA product. And now they're loaded and they've got their specific sort of target nuclease activity in there. And so the advantages, like I said, was there's no RNA inputs that are required. There's no requirement for a PAM site in your target, which is a restriction that most CRISPR diagnostics face. And then also a very straightforward multiplexing. And so, like I mentioned, the system can be used using cis and collateral cleavage. So it works as conventional Cas protein or Cas diagnostics work. But it can also target a non-functional reporter protein through a mechanism that we called Repair. And so basically the guide RNA from here cleaves out the non-functional portion, and you get a ligation, in vitro ligation, and your repaired reporter. And now you can make proteins that are, color-based proteins, fluorescent or electrochemical outputs like the glucose that we mentioned from trailase. And so here's some examples. So here we're using this system to just examples of sort of how you can multiplex different outputs. So blue fluorescent protein, green fluorescent protein, red fluorescent protein, and then also trailase, where the output was measured by the glucose meter. And then sort of emphasizing the capacity for multiplexing. Here you're looking at the red channel data, green and blue channel data. And then along the bottom are the CRISPR RNAs that were added to the different tubes. So here, the CRISPR RNA that encoded RFP Repair generates a red fluorescent signal, but not green or blue. This one, so it generates a red, but not green, but blue. And then of course, like RGB, red, green, and blue. So it was a really nice way to sort of bifurcate outputs. And then Margot applied this to the detection of pathogens in honeybees. And so here, as you know, right, honeybees underpin a lot of the food that we eat. And they're under a lot of stress right now from colony collapse disorder. And this is caused by both environmental and pathogens. And some of the most important pathogens are Israeli acute paralysis virus, or IAPV, or deformed wing virus. So we teamed up with Brock Harper from Purdue, who is a bee researcher, to build something that beekeepers could screen their colonies for these viruses without PCR. And again, it uses that same sort of barcoding idea to generate a repaired lac-C protein that you can see by eye. And so for detecting DNA, we could get down into the low attomolar range with this workflow, and for RNA into the mid femtomolar range. And then we went to look at, so here we've got the data for synthetic RNA on the left. Just check my time. Yeah. So you can see the assay could distinguish from, DV sort of either it could easily distinguish the correct sequence in the synthetic targets, and then in infected pupae. So these are whole animals or juveniles from the colonies. For DWV, we could detect DWV clearly, but then for IAPV, it's so pervasive that even the cultured, the DWV is so pervasive that it's even in the animals that were infected intentionally with IAPV. So both signals were there. And while we only looked at 19 pupae, the accuracy was, compared to RT-qPCR, was above 90%, which we were, again, pretty happy with. So at this point, I'm going to shift gears. We're almost done, to area in the lab that we're also doing quite a bit of work in, around on-demand cell-free manufacturing for diagnostics. And so our first work, this was done during the end of my postdoc with Jim. Here we freeze-dried, again, cell-free systems and DNA so that they could be shipped without refrigeration. And then upon rehydration, transcription and translation kick on, and you can either create lab reagents or therapeutics. Of course, the regulatory bar for these therapeutics is going to be challenging, but that's what we're here for. But anyways, we're nowhere near putting these into people yet. But the idea is to really distribute the capacity to have these products that are typically only available in urban settings. In this original work, we synthesized 4 protein subunit vaccines, 15 single chain antibodies, the violase synthetic small molecule synthetic pathway, and 10 antimicrobial peptides. And we took one of the subunit vaccines, the DT5 antigen, which is from diphtheria, and we tested it in animals to see if there'd be an immune response. And so on the y-axis, you have the anti-diphtheria IgG concentration in the blood of the mice. So we had our injection control mice, which of course did not create any response, or did not have a response. Then we injected a product from a cell-free reaction that did not contain the DNA encoding the DT5 antigen, and then injected the purified product from a cell-free reaction that did contain the DNA containing the DT5 sequence. And you can see that the immune response came up and peaked around 7 weeks or 6 weeks. And so this was done using Pure Express, which is a commercial system that's fantastic, but not very inexpensive. And even using that system, we could make human doses for about $10 a dose. For production, our lab's now really shifted towards lysate. So this is E. coli, lysate-based systems. I think these are the systems that are most broadly used by the community. You can make them in-house. They're highly productive, and relatively inexpensive. But they do have their challenges. And so one of those challenges is that they contain nucleases, which these exonucleases chop away at linear DNA. So we need to use plasmid DNA, which is fine, but ideally you want to be able to go from your DNA synthesis provider, do a quick PCR and straight into synthesis. And so Masood came up with the bright idea of, right, so you got the exonucleases will just destroy your protein, and he came up with the idea of having this DNA binding protein that has very, very high affinity for this DNA sequence called TER. So the protein is called TUS, and it caps the ends of your linear expression template. And the idea is that it will protect the ends. And so, and Sabina, who is a PhD student in the work in the lab, also helped make this happen. So here you can see that with increasing amounts of TUS protein on linear expression templates that had the TER sites, you can see how those TER sites become occupied. And here's just a linear expression template with 2 TER sites at each end, and the 4 products that contain TUS bound to them developing here. And so here, if you have this linear expression template with TER sites, and that's labeled fluorescently with no TUS, you can see it's rapidly degraded by exonucleases in the lysate. But in the presence of TUS, you can see there's a sustained presence of that DNA in the system, allowing you to get protein expression and transcription. So there are other systems like GAMS that work well, and others. But this is a new tool in the toolbox for, and of course there's plasmids as well. But using the TUS system, it was competitive with GAMS and plasmid based expression. Okay, so sort of to wrap it up, our sort of research in places like Toronto, where we're urban, we have relatively enough resources here. The system is that there's biotech centers around the world that ship reagents like enzymes and antibodies to use end users. And this works great when you're in urban centers. But during the pandemic, we saw that fell apart with supply chain issues, or could. And this is a challenge that researchers in low and middle income countries face all the time. It takes months to have your enzymes delivered or products delivered. And it costs 2-3 times as much because of the shipping and tariffs involved. And so we and others envision sort of an open source network where researchers can make their own reagents using open source DNA libraries and cell-free systems. And so this is something that we're pretty passionate about. And another project that Masood developed was an alternative to ELISA. And so ELISAs are really useful when you want to track, in this case, the demonstration is tracking patient antibodies, which can give you insight into natural immunity through infection, and also efficacy of vaccination campaigns. And ELISAs are great. They're very reliable and useful. But there are challenges in certain parts of the world for accessing these reagents. And also, they can be a bit laborious with all the wash steps and stuff. So Masood had this novel idea of spotting patient samples onto our serum, onto nitrocellulose, and then building a system where he had the antigen of interest that had a SPI tag on it that could be made in cell-free systems. So this is the target specific unit. And then a universal reagent that was made in E. coli that had the SPI catcher, which forms a covalent bond when it sees the SPI tag, and Apex, which is a peroxidase. And this system together can identify patient antibodies in these spotted systems. And so then, just like old school sort of development of photos, you just literally dip your membrane in solution, wash, dip, wash, and then you get your signal. So it's a very simple approach. And Masood was using cell-free systems able to develop these SPI-tagged antigens for SARS-CoV-2, the nucleocapsid protein for SARS-CoV-2, and the RBD for Ebola. And then I tested these in our lab and they worked well, and then shipped them to a third party to get testing there. And here you have SARS-CoV-2 patients. This is being probed with the RBD, I think, or no, nucleoprotein. Yes, nucleoprotein. And you can see that we were able to detect or discriminate negative from positive in almost all of the cases with the positive patients. And then hamsters that were also, in fact, sort of vaccinated with the N protein. Here we've got our negative controls, positive control, and then the positive hamster sera. And you can see it performed well here as well. And then, of course, not every lab is able to make their own cell-free systems. And so for those labs, another company called Librum was a trainee-led startup from the lab by Aiden and Alex. And they now sell lysate-based systems called Juice. And you could sort of think of it as anything you might make in E. coli, you probably can make it in cell-free lysates like Juice. And then here's some just examples of demonstration proteins. Okay, so in the end, the lab's really dedicated around making things that are generally only found in centralized settings like hospitals and labs, deployable as diagnostics and reagent manufacturing. And I've got lots of people to thank, especially Masood, Margot, Jim, Alex, Jefferson, Livia, Sarai, Larissa, Lindemar, and Varsovia, Constancia, Evan, et cetera, et cetera. So thank you all, and happy to answer any questions that you have.
Such a stunning talk and so many really exciting applications that you have going on. There are already a couple questions in the chat for you.
Oh, great.
And then just for anybody who might be watching this later on YouTube, if you could read those questions out loud before you answer them, because they don't have access to that. And then an invitation to anybody, keep posting your questions in the chat, or unmute yourself and ask a question the old-fashioned way.
So do you want me to read the questions?
Yes, if you could, that'd be
Okay, I can do
wonderful.
that. Okay, got it. Okay, so I'll start at the top. So Harvey asked, the toehold switch or toehold detection is very impressive. What's the matrix for the detection assay? Is it saliva, blood, or do you need to purify the sample? So, so the answer to that is it's complicated. So we can detect, we have detected from nasopharyngeal samples, from gargles, and from sera. And it is easiest to detect when that RNA or nucleic acid has been purified first. But we've also demonstrated we can detect by just simply diluting the sample by about 7-fold, so that you can skip the purification step. So I would say that for portable point of care diagnostics, point of need diagnostics, sample prep is probably the biggest outstanding challenge for the field. So if you can solve that, you'll be rich and famous. Okay, so Joseph asked, did you think of, do you think of an application of this detection platform for industrial applications for bioreactor contamination? Or do we have, absolutely. I think there's a lot of opportunity for using these systems for biomanufacturing, for tracking contamination, and would be super useful. Yeah, we have not done that, but I think it's the kind of thing that if you could run this instead of PCR, it might be a lot more deployable into settings like for manufacturing. And Tae-suk, hello, thanks for your question. So he said, I could not get the relation between enzyme activity and the typhoid, or the enzymes activity and COVID. I'm sorry, I did not mention that. I should have mentioned that. I skipped over that. Let me explain. So, so when the toll switch recognized the pathogen's genome of interest, that hairpin that I showed linearized and allowed translation of the reporter enzyme. And so that reporter enzyme was either LAC-Z, which could create that yellow to purple color change that you saw with your eye, or an enzyme like trailase, which would generate glucose from an inert substrate like trehalose. So does that answer your question? No.
So let me ask question directly to you. So I'm very familiar with your work. I'm a big fan of your work. I also use toll switch myself. I also published some paper about toll switch. So my question is, I get the you express enzyme, but I don't get the relationship between your enzymes and then the specific disease, because you said some compound convert into glucose and then you detect glucose to detect disease, but
yeah,
you could get those compounds from any other related situation. And also, how could you say you detect glucose that related to COVID,
that
not
is,
to
yes,
typhoid? So that's my
right.
question.
Okay, I get it. Yes, good question. So absolutely. So I didn't show it, but so there's a few answers. So if I just focus on the glucose challenge, right? So the point is that basically glucose could be brought with the patient's sample, like in the sera, and then would that be a lead to a false positive? And so the solution, so absolutely it could. And so the solutions are purify the nucleic acids, right? So that'll definitely get rid of your glucose. That's one. The next one is dilute. So if you diluted your patient sample, which we probably need to do anyways because of just inhibition from sera for molecular systems, you'd be below what we're detecting on the glucose meter. So that's like this. And then the third one is Evan also actually came up with a really clever method using the enzyme. I think it was, oh, the enzyme's escaping me, but basically it was dependent on NAD. So for every molecule of glucose it would destroy, you needed an NAD molecule. So you could destroy a very designed amount of glucose. So most glucose in a healthy person is about 10 millimolar. So if you added 10 millimolar NAD, this would basically subtract that 10 millimolar glucose from the background. And so, but you're right, we have these three sort of solutions to your challenge, but you're right, it's definitely could be a challenge if in some scenarios, but we tried to mitigate using those strategies.
So let me rephrase my question. So in my mind, the using glucose as proxy of the whatever disease is indirect method, and then potentially has a lot of problem in terms of specificity. But your method, definitely just use RNA of typhoid, the bacteria or COVID, sequence or whatever, and that's more direct without any problem in terms of specificity. So I wonder why you chose glucose as proxy to detect. Because, for example, if you have patient has diabetes that typically has a lot of glucose, and you would have the very,
so you could
problematic solution.
The other answer there is you would have the baseline, right? So you'd have like the patient's glucose level, that would be your input amount. And then the additional glucose generated by the sensor would be your test amount. That would be glucose generated by the toll switch sensor.
Right.
And that difference is is the sort of detection difference.
I see. But my question is that could be coming from the COVID, coming from typhoid or something else. But how could you differentiate saying this is the typhoid, not the COVID, but this is a COVID, not the typhoid? I mean, that's my question actually.
So you're right, but we can detect the addition of glucose, right? So the de novo synthesis of glucose from the sensor would add to the glucose, and we're focused on that difference and that would be what you would need to in the case of just trying to pull up the figure here. That's what you would need in a case of like point of need where you have to you don't have the luxury of purifying your nucleic acids. I'm just going to share, and then we developed this tool here where so it's glucose dehydrogenase, right? And it's a mutant that when you add 2.5 millimolar NAD, you reduce it's molecule for molecule 2.5 millimolar of glucose, 5 millimolar, 10 millimolar. So you can kind of with some precision remove your glucose background with this glucose dehydrogenase enzyme as well. So it's a way to sort of subtract that patient material. So it's not a perfect system, that's for sure. But we chose glucose just because of the availability of the hardware.
Sure.
Yeah.
Okay, thank you.
Yep. And then Finu, I'm not sure if hopefully I pronounced your name okay. So is it possible that non-specific nucleases can cleave 12 switches and give false results? So the first part, absolutely. Yes, patient nucleases could chop up your system. The assay is run in the presence of a control. So if I didn't mention this, but you read it at a fixed time point, like say 90 minutes or 60 minutes or something. And so you would be comparing your treatment, just like you have on lateral flow test, right? You've got your control band and your test band. Same thing in this type of an assay, you'd have your control paper disc and your test paper disc. And so as long as your control was still negative and your treatment was positive, or your query test was positive, then but you're right, you can totally get false positives, and it has taken quite a bit of work to sort of get that figured out. That's a large part. I would say that's a large part of like my group's effort, is like mitigating the challenges of false positives. So it's a challenge for the field in general. So thanks everyone for your questions. Much appreciated.
Thank you, Keith. That was awesome. Very, very interesting. I'm going to put out the last call for questions if anybody wants to throw them out there. Otherwise, again, another final thank you to Keith and to all of our audience who are amazing every week for us.
Well, thanks for the opportunity and thanks for the questions.
So may I ask one last question?
Oh yeah.
So let me clarify, because I still want to get the real answer. So is it known to community typhoid related to, for example, glucose phosphatase increase? If that is the true, I may have the better idea. Or COVID related to increase of glucose phosphatase or any enzyme you just mentioned. That part I couldn't get the connection.
So you're saying there's
So the assumption I got from your presentation is if you have the typhoid, the 3 enzyme you mentioned should increase, right?
Yes, yeah.
But is there any report that related to those increase in enzyme if you have those disease, including COVID and typhoid? That is actually
I don't know. That's
so why you chose those 3 enzyme at the beginning? Could that connect with those disease, including COVID
we chose those enzymes as reporters. So just like you would choose GFP as a reporter for an optical output,
okay.
we chose those. Let me just
Because I probably misunderstood your system. Your
maybe,
system.
yeah. Let me just try to get to a slide that might help. Then I'll share
So
it.
what cohort switch, sense in terms of sequence? Which sequence you sensing? That's my question actually.
Right. So let's say this was for typhoid. The toehold switch would be engineered so that this region here would be specific for the typhoid genome.
Oh, so you are
Yeah.
detecting the typhoid genome here.
Yeah.
Okay,
And
now I got
sorry.
it. Okay, so that means that you need to have purification for the genome anyway.
Or you could dilute the sample by sevenfold, and then that would usually work. And then instead of GFP, you're making one of those enzymes that makes glucose.
Okay, now I got it. So I'm
Okay,
sorry.
sorry.
I misunderstood that part because I thought, you have some very unique way to.
I wish.
Okay.
No.
Okay. Now
I wish we did, but
so
no.
you basically sensing the sequence of the DNA, the RNA or genome or the DNA sequence. Basically, you just sensing those things by breaking the cell of the typhoid, the Salmonella typhi or COVID, right?
Yeah. So the glucose production is linked to the presence of the genome sequence. Yeah.
I see. Okay.
Yeah. So that's all. Yeah. Sorry for not making that clearer.
Yeah. So now I got it. Thank you. Now
Okay.
I completely understand. All right.
You bet.
Great.
Wonderful. Yeah, we do have a question in the chat asking how or if someone can reach out to you afterwards to continue the conversation.
You bet. Please reach out. I'll just find my email address and put it in the chat. Love to talk more to anyone. And so my email address is there.
All right. With that, I'll give you guys a couple seconds to copy it and then say goodbye and have a good week.
Awesome.
All right. Thank you again,
Great.
everyone.
Thank you all. Thanks a lot.
Bye.
Take care. Bye.